I may have your attention. So welcome everyone, colleagues, visitors, friends, and family of Dario. I'm really pleased to welcome you to this fantastic occasion where we introduce our newest professor to you. Dario Farina, Chair in Neurorehabilitation Engineering, joined Imperial only in October of this year. And so I think this is the fastest turnaround for the organization and preparation of an inaugural lecture. And much of that goes to the support of James Romero and the team who've done so, but also Dario. He joined us in October from the Georg August University at Göttingen, where he founded the Department of Neurorehabilitation Engineering. He is an electronics engineer by first degree, and he obtained his degree and his PhD from Turin, and has also studied in France, and has held academic positions in Turin, Aalborg, prior to going to Göttingen. I think in the couple of years that I've got to know Dario, I'd like to say that I think there are three words that describe him, at least how I would describe him. He's excellent. Of course, we will see that tonight. He's prolific. I think we might see that as well. And he is a strong collaborator. So prolific, over 300 peer-reviewed journal publications. He might say over 400 now, I don't know. The number seems to go up by day. Over 16,000 lifetime citations, that means something to some people. Over 10 million euros in research funding, which means more now to us than it did a few months ago. And numerous books and patents and invited talks. His excellence is recognized through many international prizes, keynote lectures, and awards, including the prestigious Fellowship of the American Institute for Biological and Medical Engineering. And of course, his excellence is more fundamentally exemplified by his research, about which you will hear this evening. And so finally, I'd like to emphasize that he is obviously a strong collaborator. I first met him a couple of years ago in the context of a collaboration with one of our other professors in the department already ongoing. And since he was appointed prior to starting, he has also forged many new collaborations here through grant applications, developing with collaborators for the college a research and translation strategy for neurorehabilitation. And we're pleased about that, and we look forward to the fruits of that in years to come. So we are absolutely delighted that Dario has joined Imperial College in the Department of Bioengineering, and I would like to invite him to give his inaugural lecture.
Thank you, Anthony, for the wonderful introduction. Dear colleagues, friends, family, good evening, and welcome also from me to this inaugural lecture that marks my commencement at Imperial College. This evening, I would like to discuss with you research I have been doing within the field of bionic limbs in the past. And I would like to do that without the engineering or mathematical complexities, but in a way that can be fruitful for a broader audience as it is present today. So I hope you will enjoy. I will start my talk on a general perspective. I would like to discuss with you, before going to the core to my talk, on how we scientists study human movement, because the topic of the lecture is how to restore human movement in case of pathologies. So if we look at the scientific literature, neuroscientists study human movements from the point of view of the neural cells. And we have very advanced models of the working principles of neural networks and neurons. At the other end of the discipline, human movement is studied also with the discipline of biomechanics. And this is a very different approach. It's the approach for which movement is investigated by studying the forces exchanged between the skeletal segments and the external environment through our muscles. Now, there is a gap between these two disciplines and these two ways of studying movements. And specifically, our main difficulties are in understanding the functional significance of the neural activity that actually determine function. So our main challenge is to link neuronal activity to behavior. So today's lecture is about neurotechnologies, and you can think of neurotechnologies as an area of research that is aiming exactly at filling the gap that we've just discussed. Within neurotechnologies, what we are trying to do is to decode the neural activity to predict function. And within this very broad definition, you can find basically all types of technologies on which we work for regaining and restoring movements, from bionic limbs to robotics for rehabilitation to assisted devices such as exoskeletons and endless of other applications. So the field of neurotechnologies has had very important advances in the past couple of decades. And the advances have been so important that just last month in Zurich there have been the first Olympics of bionics, of bionic medicine, the Cybathlon. This was in October in Zurich. The best research teams in the world and patients themselves engaged in competitions, in an Olympic spirit, of course. So good type of competition. Imperial College was also represented by Aldo Faisal, and actually arrived very close to the gold medal in a number of these disciplines. So we are at this point. We are at the point in which bionics and the most advanced technologies that we have are competing with real patients in elite sport type of competitions. So having said that, this was to introduce to you the topics of the lecture. I will now go to the core of the topics I would like to discuss with you. And I structured it in a form of questions and answers that I will try to give to you. There are three main questions that you can read here, and this relates to the substitution of limbs with artificial ones. So we will go from the anatomical substitution to the neural control, and then we will look at the future, and we will discuss if limbs that are surrogates of biological limbs can eventually become even better than their biological counterparts. This will be a sort of a vision for the future. And then my last question is related to what I will be doing here at Imperial College, which I think is also of interest for most of you. So we'll begin with the first question. And the first question that we address is if we can substitute, from the anatomical point of view, human limbs with robotic counterparts. What you can see here is a video representing the state of the art of leg prosthesis, above-knee leg prosthesis. This is a commercial prosthesis. The company producing this is Ottobock, but other companies have similar products. And you can see that in terms of recovery of function, this technology is quite advanced and allows patients to regain locomotion not only in overground walking, but also in very challenging conditions, such as these ones. We can study these systems in a variety of different ways. We have very good models of the human anatomy and the mechanical interface with this prosthesis. We can design them by modeling completely the function that they are supposed to replace. If we now go to the upper limb, as you may imagine, the upper limb poses more difficult challenges. And it poses more difficult challenges because we had to recover very fine dexterous control of the hand and also the reaching actions of the arm. And you can immediately see that the number of degrees of freedom and complexity of that function is much more relevant than what we have seen with a single degree of freedom in that robotic knee that you have just seen. The human hand is a beautiful, sophisticated piece of equipment, biological piece of equipment. It is controlled by more than 30 muscles. It has more than 20 degrees of freedom, and as everybody knows, allows a fit of actions from very fine manipulation to very strong actions. So can we replace this beautiful biological device with robotics, with inorganic means? Well, we have currently very advanced prosthetics that replicates the anatomy of the hand. Maybe they don't cover exactly all the degree of freedom of the hand, but allows to replicate most of the functions. And if we look at the pure robotics, we are also able to replicate extremely fine control of robotic hands and arms. Here you can see from KUKA robotics actions of a robotic hand autonomously controlled that are mimicking very complex and accurate actions of humans. But now you have to be very careful. The key word here is that we have autonomous robotics. So there is no human interface with these robots. These robots have sensors on board. It has very complex control algorithms, but there is no connection with humans. Now, when we consider the same piece of robotic and we try to interface it with humans, the situation becomes much more complicated. This is from Brown University. It's a brain interface. So now the human is controlling this robot. You can see that the hardware, the robotic arm and hand are similar to the one you've seen before, but just by looking at which are the possibilities in this case, you can see that the type of control is much less impressive than what you've seen before. So when we interface humans with robots, we find the bottleneck. And this leads us to our second questions. Are we able to interface the nervous system of humans in order to have a real dexterous control of robotic devices? This leads us to the topic of man-machine interfacing, on probing the nervous system and decoding commands that may control robotic parts, mechanical interface with the human body in a natural way, as if they were the real limbs. This is the real challenge in the field. How can we do that? Well, we can do that at various levels of the neuromuscular system. We can implant electrodes directly in the brain, and this would appear to be the most obvious solution because clearly our movements originate from the brain. It is also the most challenging one for a number of reasons. We can interface the spinal cord. We can interface peripheral nerves. We can even interface muscles. This is the full neuromuscular system. We can put electrodes or other type of sensors. We are not necessarily linked to electrical activity, and extract movement intention from humans in order to translate this to movement of prosthetics or robotics. So in this lecture, I will focus on these aspects, the peripheral aspects from the spinal cord and peripheral nerves, to be a bit focused. Now, if we think of peripheral nerves, we may ask ourselves, how many nerves do we need to decode an interface? And these are data from Oscar Atzmann in his laboratory in Vienna, from cadaver donors who have been used to stain efferent and afferent nerve fibers along the human arm. And these nerve fibers are labeled as efferent, so they give commands, and afferents, they receive commands. And when you do something like that, Oscar and I were very surprised when talking about these results, because to control the full intrinsic musculature of the hand, all our gestures with the hand, you need only around, in total, 2,000 nerve motor fibers. It's an extremely small number if you think at the number, for example, of cortical cells that at the cortical level control the action of the hand. So this is important because it means that in principle, we will be able to interface all of them. We will be able to completely decode all of them. They are not in such a large number. We are not talking about millions or billions, actually, of cells. We are talking about a few thousands. So that's a very interesting characteristic of peripheral nerves. And remember, if we are able to decode the activity of all these nerve fibers, we have the full information about the function of the end. So nerves are also interesting because we can interface them in smart ways. So interfacing nerves implies, in principle, to put electrodes into nerves themselves. This is a complex procedure, though, for many reasons. And also it provides signals that are relatively poor in terms of quality with respect to noise. But nerves have the properties, being in the periphery, that they can be rerouted to innervate biological tissue that we can use as a biological amplifier of the nerve activity. So this is a surgical procedure originally developed by Todd Kuike in Chicago, and then refined and modified by Oscar Asman in Vienna and by others over the past decade, that consists in surgically taking nerves that would normally innervate a missing limb and reroute them in muscle tissue. So that muscle tissue is a biological amplifier of the nerve activity. And then we can interface muscles in order to decode the nerve activity. That's a very powerful solution that implies, of course, surgery, electronics, and eventually robotics when the patient is fit with the prosthesis. Here you can see an example of patients interface in this way. So the colored map that you see are muscle activity, but the muscle activity is the amplified nerve activity of the nerves that were innervating that missing limb. You can see how strong is that activity. So the biological amplification is very powerful. We are able now to decode, depending on the movement that the patient is attempting to do, to decode the nerve activity in all these points of the skin by recording from the muscle fibers that are amplifying those nerve fibers. All right, so once we reinnervate nerves into muscle and we use muscles for biological amplification, now we have to put into it some decoding. So at this point, we have a so-called reverse engineering problem. We know that what we are seeing at this level is the electrical activity coming from the spinal cord. And now mathematically we want to disentangle that electrical activity in order to obtain from a peripheral recording the same information coming out from the spinal cord. This can be done with advanced muscle interfacing. It can be done by placing electrodes, high-density electrodes over muscles. And here you can see modern systems of this type. You can place hundreds or even thousands of electrodes over muscles. And here you can see, by the way, the Mole Antonelliana from my own town in Torino. This system had been pioneered when I was mentored by Roberto Merletti, who is here, some years ago, as you can see. And they allow to record the muscle activity over a number of locations on the skin surface. We can also interface muscles reinnervated by nerve with electrodes implanted into them. And this is another place where I spent quite a few years of my career in Aalborg University when I was professor there. This is Ken Yoshida, the colleague of mine, with whom we develop microfilaments to insert into muscles and to decode from, in principle, tens to hundreds of points within the muscle. Here you can see a beautiful picture of the Aalborg Fjord in the north of Denmark, and this is the Utzon Center, dedicated to the architect Utzon, who is originally from Aalborg, and who is the designer of the Sydney Opera House, who is more famous than this building, but this is also a very nice building. Well, this is the type of interfacing that we have developed over the years. And then we have also developed decoding techniques. So with this interfacing, as I said, recording from muscle that are amplifying nerve activity, mathematically we go back at this level. We decode the output of the motor neurons in the spinal cord. So putting together surgery, mathematics, electrodes, now we have an interface with the spinal cord that is viable in patients. It requires minimal implantation techniques. Of course, it requires surgery, but it is very stable and very robust. What you can see at this barcode is the full neural code of a voluntary movement decoded in this way. So each of these small lines are action potential of axons from motor neurons in the spinal cord that have been decoded in that way. I dedicated the past 6-7 years to develop the mathematics to obtain this. And this happened when I was directing the Institute of Neurorehabilitation Systems in Göttingen University, and especially within an advanced ERC grant that really helped us to boost a lot these activities. Here you can see a photo of the center of Göttingen in a summer day. That's also a beautiful city. All right. The interesting thing now is that we can put together all these concepts that I presented that are laboratory concepts. This is basic research, but it is possible then to translate these concepts into patient solutions, into technology that can be used by patients. And this is an example of one of the most advanced ways to do that. What you can see here is a radiography of a transhumeral amputees. This is Oscar Asman. He's not the amputee, of course. He's here, and he's the surgeon who has treated the amputee. And in this picture, you can see the full exploitation of the concept I explained to you. These patients have received surgically a nerve rerouting into accessory muscles, and these capsules are implanted EMG electrodes that have been inserted very close to the reinnervation points. And they transmit with inductive coupling through a coil placed externally to control a prosthetic hand. So you have all the concepts I presented to you now translated in a system that is used on a daily basis by amputees. This is one of the most advanced treatments we can employ on amputees currently. And this is how a patient utilizes these systems. This is a prosthetic hand, as you can see, and it's still in a laboratory. This is my ex-postdoctoral researcher, Janne Anne, in our laboratories in Germany. And with the interfacing that I've explained to you, you can see that the patient is naturally controlling that robotic hand. Now, the difference with respect to the initial video on the autonomous robot is that here you have a symbiotic connection between the robotics parts and the human. And the human is controlling these robotic hands in a way which is natural, and in a very similar way that he would do with his natural hand. These are the laboratories of Oscar in Vienna, and these are more clinical tests. You have the same patients actually in this case. And this is an example just to show to you of the type of clinical test we employ to evaluate the efficacy and effectiveness of the restoration that we can provide to the patient. And in particular, you can see that the patient is able to perform quite delicate tasks with his hands. So we believe that this is among the most advanced technology for amputees, or for patients in a translational way, so that amputees can use on a daily basis that is currently available. So we control machines, but we inhabit our body. So for patients, it's important to feel that part, and not as a tool, but as a part of his body. And here you have subtitles of what the patient is saying as a reaction when he was trying this system for the first time. And one of the lines that he is stating, as you have seen, is that's my hand. So he feels that as his own hand. He feels that his own hand because the control is so natural and so immediate and accurate that for a simple task, we are not talking about very advanced manipulation, it can control itself basically on par as a biological hand. In Germany, we also had a lot of initiative of presenting to the public this system. This is our friend and colleague Patrick, who is an amputee, as you can see, and he is showing and explaining to people how he controls his hand. This is one of the largest public fairs in Germany on technology and related topics. So far, I have presented to you a way of interfacing the nervous system of humans with robotic hands that is basically unidirectional. So it goes from the brain of the patient up to the spinal cord, and then the nerves are interfaced, and the prosthetic hand is controlled. But remember how advanced it was, the autonomous control of robotic hands in one of the initial videos that I've shown to you. So the message of that video is that we can put additional intelligence on the robotics. We can make the prosthetic more intelligent and actually having a bidirectional communication with the patient. That is what we did, in particular in collaboration with Ottobock. This is Bernard Greiman. He's one of the technology managers of Ottobock. And the idea here was to equip the patient with smart glasses or other type of smart technology that could help performing tasks. I will not go into details, but I will show you an example of this effort. This is a patient who is wearing Google-type glasses or similar type of glasses. And these glasses are cameras that can analyze the scene. And behind that, there is a lot of computation that the microprocessor is doing. This is doing in real time. Here you see it slow down. So what the microprocessor is doing is to get information about the environment. The system can see this object, it can model these objects, and it can help the patient control in the end in an optimal way. For example, if an object has a particular size, the robotic hand can open automatically for that particular size, and then the patient can still correct that if he's not happy. So now you have a full symbiotic relation between that robotic parts and the patient in two directions. So can the human brain control bionic limbs? Well, then the answer, yes, we can do that. We can do that not only in the laboratory, but we can do that with patients. We can provide to patients good solutions that can partly recover the function. It can be done better, and this will be, of course, what we will do in the future years, but the technology is at the availability for real patients in a translational way. Now, having seen that, and having seen these videos, one of the questions that may come into mind is, well, if we are at this level now, will we be in 50 years at a point where a robotic arm would be better than a biological arm? Why not? It may happen even before. We don't know. But what will it happen if that is the case? Which kind of bioethical issues would be raised? Well, at the moment, this is not possible, meaning that prosthetics that you have seen, they are very advanced. They provide very impressive control capabilities and very impressive tasks, but I'm sure that none of you would decide to have his hand chopped in order to get a robotic hand. Well, what I will show you now, and this is the last of these three questions that I have, are actually a few individuals who made exactly that decision. And these individuals are particular. They had not a fully functional hand, but they had hands that were fully anatomically intact. They are patients with brachial plexus injury. These patients have an injury at the nerves innervating the arm, and this injury implies that they have difficulties in controlling their arm, usually with physiotherapy, with surgery, and with a number of biological solutions that they can recover the function of the shoulder and the elbow, but they have often hands that are not functioning at all, as you can see. They don't have sensation, and they cannot be controlled. So some years ago, Oscar had at his door a number of these patients asking him, as a plastic and neurosurgeon, what to do for them. These patients may be in that condition for 15 years, and they have an appendix that is completely useless. And the answer from Oscar is that all the biological solutions were not available and not possible. Everything had been tried. And so what he did was to set up a symposium in which he invited a number of people from many different disciplines, even theologians, experts in ethics, patients and surgeons. And they discussed for around 1 year on what to do. And what they discussed specifically was a very brave proposal. The proposal was to remove that hand that is perfectly intact but is not functioning, and to substitute it with a robotic one. Now you can see that this is very much related to the question that I posed you, because this is obviously not a perfectly working hand, but it's a step forward with respect to the case of patients that have had a trauma and they don't have an hand. In this case, the hand is present. So conceptually, is an important step. Well, the answer from this was that it was probably ethically appropriate for these patients to provide and to suggest them, who of course have to agree, this solution. And what has been done here was the combination of the most advanced surgery, electrodes techniques, and decoding algorithms in order to provide these patients a new hand that could be controlled in a better way than their existing hand. In particular, the damage at the nerve implies that even getting signals from the nerves with the techniques I've indicated to you was challenging, because these signals do not actually come down. So what was done here was a transplantation of muscle tissue, small amount of muscle tissue. And the reason, you may understand for what I have discussed before, was to provide a biological amplification of the nerve activity. So more sites from where it was possible to probe the nerve activity. And then these patients have been equipped with the best available interfacing, and at the end, their hand has been substituted with a robotic one. So what you can see here is one of these patients before this procedure. You have seen a similar video before. So this is his natural hand. And this is after 1 year and a half with his robotic hand. So his natural hand has been substituted, has been interfaced, and the function of this robotic hand is far superior than the original one. This is another of these patients that is performing a daily living task. It's not a perfect hand, but with respect to what these patients could do with their natural hand, this is a very big improvement. And in fact, these patients are extremely happy of this solution. After now it's a few years since this solution has been implemented in them, and they are extremely satisfied about that. So you can see the same patients doing very accurate tasks. So not surprisingly, these scientific reports attracted a lot of media attention. And the media attention is always, as classic for media, aimed at emphasizing the appealing aspects. And of course, the word cyborg appeared in many occasions. But the interesting aspect here is that is actually coming back to the question that I posed to you at the beginning. So, will the robotic hands eventually become better than our own hands? This is not the case, because we are in a very particular pathological condition. But somehow it's very appropriate to provide you this example, because these are the first individuals who decided for a bionic substitution of their natural hands. And if you think at the implication, as I said, from the ethical point of view, imagine that in some years from now, people could get better hands, then who decides who gets what? Who pays for that? So we've been discussing over the years this question with the ethics department in Göttingen, for example, and people making research in this bioethical question were extremely excited by these results, because this opened a completely new possibility for discussion at a bioethical view. All right, so this was a presentation, as I said, without mathematical or engineering details of a few core questions in bionic limbs. What I would like to do now is to discuss with you which are my plans for the work that I will be doing here, and which are also the plans that I've been discussing with many people here, and where I have found a very, very large enthusiasm for these ideas. So many of you will know, but not all of you knows, that Imperial is building a new campus at Y City, and not all of you know that biomedical engineering will have a major role at this new campus. What you can see here is a computer rendering of the Michael Uren Biomedical Engineering building that will be built within a couple of years from now at Y City Campus Imperial. And the strategy is to host in this building a number of areas with this biomedical engineering, among which neurorehabilitation engineering is one of them. And I have the honor, together with other people, to organize these activities in view of the availability of this exciting new campus and a new building. So our aim, our ambitious aim, is to found an institute that will deal with the topics that I've discussed with you, so mainly translational topics, topics that goes from basic research to patients, to real patients, not only within bionic limbs, but also in a number of other topics on which I've been working in the past. And this basically would cover all the aspects in neurorehabilitation technologies, from brain stimulation to robot-aided rehabilitation, biomechanics, of course, prosthesis, and robotic limbs. So we are ambitious, and so we would like to set our goals very high. And our ambition is to found the London Institute of Neurorehabilitation Engineering at the White Campus of Imperial. And in our view, this should be a world-leading institute for the design and clinical translation of neurotechnologies for rehabilitation at the frontiers of neuroscience, robotics, and medicine. And I believe, and many of us believe, that Imperial College is worldwide one of the best places where to attempt an initiative like this. With this, I will conclude my talk. I would like to thank you all to being here. Thank you, Anthony, for making this inaugural lecture possible in a short time, and for all our discussions, and for having me here as professor at Imperial. I hope you enjoyed the lecture. I hope it was not too heavy for this time of the day. And especially I wish you an excellent continuation of the evening. I hope we can interact socially following this lecture during the reception. Thank you very much.
So for those, I didn't introduce myself at the start. So my name's Anthony Bull, and I'm head of the Department of Bioengineering, so just in case you wondered. My role is not to thank Dario at this point, but I really will want to, actually. So I will do so later. Okay, but we do have a formal way of doing this, and it's called the vote of thanks. And it really is my pleasure to introduce Professor Roberto Merletti who has worked with Dario for many years. In fact, right at the beginning of his research career, since 1997, I understand. As the founder and director of the Laboratory for Engineering of the Neuromuscular System at Politecnico di Torino, he has produced and led groundbreaking work in muscle physiology and electromyography, EMG, and you've seen some of the fruits of that here today. So he is not only a good person to give the vote of thanks, he is the perfect person to give you the vote of thanks. So I'd like to invite Professor Melletti here. Fantastic.
Thank you. Thank you very much. Thank you very much for the nice presentation. I am greatly pleased and deeply honored to be here today. And I would like to start from the beginning. I will tell you a few things about Dario. I will say only the truth, nothing but the truth. This is the first page of his PhD dissertation, which was supervised by two supervisors. One is shown here to the right. The other one is now skiing somewhere on the Alps. Professor Christian Doncarli is a great expert in signal processing. And he was also one of the important trainers and people and supervisors of this work, but also people that conditioned to a considerable degree the work that Dario did later. The love for mountains is shared by Doncarli, by myself, and also, but of course to a much greater degree, by Dario. So doing research, as you can read, is like climbing a mountain, it's overcoming difficulties, solving problems. This takes hard work, and it takes a lot of difficulties. It takes standing cold weather and climbing hard slopes and snow. So that some people can do one thing, some people can do the other thing, some people can do both. And in this case, with a lot of sweat on, because in this building that you can see on the right, the heating was not really strong. And this is at 5,000 meters above sea level, where two of my collaborators that you can see there spent some time in studying the effect of hypobaric hypoxia on muscle signals. This was in 2002, 3? 98. This was in 98. See how time goes by, very fast. So here we have some pictures of that adventure. You can see a yak on the right, carrying a big bag with our own equipment that Dario contributed to build. And although this equipment was completed a few hours before leaving, actually it withstood 5,000 meters of altitude and the shaking of being carried on the back of this animal. And this is the Everest range that you can see on the left. Now, I don't know whether Dario's mother has ever seen this picture. I hope not. So this is a tricky passage, like you have often in research. Some difficult steps that you have to face and that you have to overcome and to get on. And at the end, despite all these difficulties, often, not always, but very often, you get your rewards and get to spend some time with good friends, nice ladies, good wine. Now here you may notice that there are a couple of water carafes that have been placed artificially there after removing some of the empty bottles. Now, to get a little more serious, I would like to make a statement that is the deepest, the greatest reward for a teacher is to train people who surpass him. I don't know who said this, but it's certainly some famous philosopher, probably Oriental. And so I have been very lucky, and I have been very lucky to be able to come here and tell this to you. And so my best wish to Dario is that he will be able to say the same thing in many, many years to come. Thank you very much.
To thank you all for attending. We can now retire for a glass of wine, and you're all welcome to not only socialise, but perhaps really probe the answer to some of the questions that Dario has only half answered today, I think. So thank you all, and thank you especially to Dario.